very much welcome. Today we're going to look at support and transport in plants. We will be looking at all the different tissues that make up the stem and the root, and then we'll be looking at how the water flows through all of these different organs. So let's start by getting right into it. Alright, so this right here is the internal structure of a dicotyledonous root. Right, you can also just refer to this as a dicot root. And this image right here, this diagram, you need to be able to identify as a root. Okay, we'll look at the stem later on, and they look a little bit different, and you need to be able to differentiate between the two. So what makes this a root? Very obvious is there are root hairs, right? And then we have an epidermal layer, and then there's parenchyma tissue and endodermis, and they make up the cortex. And then there's a central cylinder, which is made up of the pericycle, the phloem, and the xylem. And we'll be looking exactly at each one of these functions and specifically how to identify these tissues. So zooming in on that picture, we're going to look at the cells a bit more in sort of as zoomed in image, and there you can clearly see the root hairs. Here you can see the parenchyma cells that make up parenchyma tissue, and there are even some gaps in between the parenchyma cells. So if you've done this at school, you've learned that parenchyma tissue is made up of these big cells, and in between these cells there are these gaps which we call intercellular air spaces. Then next up we have the endodermis. This plays a very important role, which we'll look at a bit later on. And then there's a layer just below that known as the pericycle, and then the xylem is in this X structure, and then phloem is in between the arms of these structures. So we'll be looking at each one of these tissues individually and their functions and what role they play in a plant. So this is just a picture of a carrot, which we know is a root, and here you can also clearly see the cortex area in this diagram and the cortex area of the carrot. And then here in the middle we find the central cylinder, which is on the left hand diagram over there. Right, I'm not sure if you can clearly see, there should be like this X in the middle or a bit of a darker area, and that would then be the xylem, and in between would be the phloem. So diving into the first sort of layer is the epidermis. You've learned that the epidermis is the outer protective layer of this root. It's also transparent. In the epidermis of the root doesn't have a cuticle. That is quite important, and that makes it easier for water to be absorbed. Right, and then the root hairs, as you learned in the previous chapter, their function is to make the area it increases the surface area for absorption, so that water can be absorbed throughout this area, and then there's another area. So there's this larger area from which water can be absorbed. It's not just through this one sort of root in the middle. And now Yolandi will just take us through the cortex area.
Right, so in terms of the cortex, we've now entered, let's say for instance we're a little water molecule, we've now entered through the root hair, we've crossed over the epidermis, and as Monica mentioned, we're going to now move through that cortex section. So the cells that make up our cortex is called the parenchyma cells. Now, the way in which I remember parenchyma cells is I think, okay, parents, you know, and then I draw a picture of two parenchyma cells next to each other, but I give them personalities. So I draw like a stick figure, and two round circles represent the bellies, okay? And then it's two, the mother and the father holding hands, and they're so fat. Let me quickly draw a picture just to get a visualization of it. So then these will be the bellies, okay, and little feet, okay, of the individuals. and they're holding hands, and this is now the mother and the father. Okay, so imagine that's a little ponytail, and that's the father. Okay, so I give it personalities, and I say that this parenchyma tissue, and I make the association that it's parents. They are so large that when they stand next to each other, their bellies touch each other. Okay, and then I think, okay, what can I associate with the bellies? And that is where food is stored. So now parenchyma cells are large cells. Okay, there's space in between the cells, all right, and their role is to store food. So by making these little association diagrams, I'm able to make a link with what is the function of a parenchyma cell. One of the functions is that it's going to store food, and that will obviously be your starch that gets stored inside roots. Now, we know things like carrots are nice and sweet, and that comes from the fact that that glucose is actually stored within the root structure itself. Right, so now if we take it a step further, in our diagram, obviously, if it's a water molecule that's now being absorbed from the soil, it's going to pass over the parenchyma cells, which are thin-walled, and that makes it easier for things to move across, of course. Okay, so then the intercellular spaces are present within this layer as well. Yet again, that intercellular air spaces allow for easy and fast diffusion of substances or osmosis of substances. And eventually we're going to get a layer called the endodermis. So in a test or exam situation, you might need to explain the pathway of water, and then you start with the root hair, you move over the cortex consisting of parenchyma tissue, we reach the endodermal layer, which is this section here, and right after that we will reach the pericycle and the vascular tissue. Right, so in terms of the endodermis, what we will find in it, it's tightly packed parenchyma cells, so no longer does it have those intercellular spaces. They are densely packed next to each other, but we do have a special type of cell that's present here, and this is called a Casparian strip. So these guys are waterproof strips that actually redirects the flow of water. So normally what we find is that water can take three pathways. It can either move through the cytoplasm of a cell, so straight through a cytoplasm, or it can go vacuole to vacuole to vacuole, or what it can do is it can go between the cell wall and cell membrane. Now that Casparian strip will redirect the water so that it enters into the cytoplasm before it can actually reach the vascular tissue, which is in the middle. And that vascular tissue would then be your xylem and your phloem. But if we're talking about water, the end goal of the water would be to get to the xylem, of course. All right. So in terms of our diagram, the Casparian strips control how the water moves through it. Okay, and this can be water or nutrients in order to reach the end goal, which is that vascular tissue. So it's not ideal for a plant just to take all the water that's surrounding the root and just absorb it in one go. We want to pace how the water is entering this our vascular tissue. And the Casparian strip kind of plays a role in almost like being a traffic cop and ensuring that the water is moderately taken up from the soil. Okay, so there's our cortex yet again. We can see those nice air spaces that look like triangles between our parenchyma cells. And then obviously in terms of making the water move, that allows for that fast diffusion that we were talking about. Also important for storage. We spoke about the glucose being stored as starch within it. We also find that there's a lot of water that's within this section. So that when we cut into potato, usually there's a bit of water that comes out of it. So that gives us an indication that plays a role in storage. Okay, and then we have our endodermal layer. We can see the black dots there. Those indicate our Casparian strips. And then we reach our pericycle of the root. Okay, so this is now this darker band that we see just before we get to our vascular tissue, which is our phloem and our xylem. Now, a lot of grade 10s, they struggle with remembering what is xylem and what is phloem. I've got a very silly way of learning. So when it comes to things like phloem, I think, when I struggle to spell something, food is with F, but then sometimes you get that same F sound, and then it's usually spelled with a P and a H. So I go and I spell food deliberately different in that I write down food as PHOOD. And then that's the association that I make with, okay, the one that transports things like glucose and sucrose and all of that, that will be your phloem, food, okay? And then the other one will obviously be for the transport of water. So that's just a little bit of a differentiation that I take when learning the different functions of the structures. Right, so let's quickly hop into our diagram. So now we've indicated where that central cylinder is located in the blue, and all the components that it consists of. Right, so it consists of our pericycle, which is the black band. It consists of the xylem. X marks the spot, like Monica mentioned earlier. We want to see an X-shaped appearance to it. And the spaces in between the X, that will be your phloem as indicated in the picture. Right, so the function of it plays a role in support and protection of the vascular tissue, so it keeps it nice and open, and it produces that lateral root section as well. Okay, so in terms of the xylem, it's recognizable by being in an X shape, as we can see here, and then as indicated earlier, the phloem will be in between the arms of the letter X of that cross. Okay, so the rest of the cylinder consists of parenchyma cells, and their function would be to store starch, which is obviously glucose that's in its storage form. Okay, so just an image to show us a close-up video of image of it. Here we've got our vascular tissue in the middle, and here you can see the definite X structure in that darker purple color. Now the lighter blue that we see on the sides, that will be a phloem, and we can almost make out a pericycle surrounding it. In this image, it's a little bit more clear. This darker section here indicates our pericycle. Here we've got the xylem, and in between it, we've got our phloem. Right.
All right, so now that we've done the root, we are going to start by looking at the stem. So this stem looks, there are some similarities between the stem and the root, but there are some differences. So as Yolandi mentioned, the root has that X marks the spot in the middle, whereas the stem has these sort of Easter eggs that encircles the this middle part of the stem. And these little Easter eggs, they aren't Easter eggs, they are vascular bundles. So they contain our vascular tissue, the xylem and the phloem. So we'll be looking at this stem. So looking at this, you need to identify that. And we will just quickly zoom in on one of these vascular tissues, or vascular bundles. So zooming in on the structures of a stem, there's you can also have some hairy outgrowths. So just by looking at these little outgrowths, you can't just assume that this is root hairs. Some stems also have some hairy outgrowths. Then we find something known as the cuticle that is now present on top of the epidermis, and that basically just prevents water from being lost. So then there's a layer that we haven't found in the root, known as the parenchyma. And as you can see, these cells are extra sort of thickened around them. Their cell walls are evenly thickened, and that contributes to some extra strength and support for the stem to not just be blown away by the wind. There's parenchyma again, and then this little bundle over there is also then made up of different parts, which we will also be looking at in detail. But just for now, there's this endodermis layer again, then there's the sclerenchyma cap, then we have the phloem, and then the cambium, and then we have the xylem. Alright, so for water to move into the stem, this xylem and phloem, or xylem transports the water. It needs to basically go from the root into the root's xylem, and then it will be transported up to the leaf through the stem's xylem. Alright, so looking at this, the first part of the root of the stem, we again have an epidermis, and how the epidermis is different from between the root and the stem is that the epidermis will now have this cuticle. Alright, and this cuticle is sort of a waterproof jacket that the stem has on, and it prevents any water from getting lost. Some cells can even have some stomatas, but you should know that stomatas are mostly found on the leaves, and these cells are again just single layered brick shaped cells, but they, in this case, they have that cuticle. Then moving over to the cortex area. So the cortex is made up of parenchyma tissue and then also parenchyma tissue. Oh, and then also the endodermis. Right, so those are the three parts that make up this cortex. And we'll just quickly also be looking at their individual functions. So the function of the parenchyma, as Yolandi mentioned with a cute little example, they have these intercellular air spaces. And in this case, we don't have water needing to move through the stem. These air spaces are there for gaseous exchange to take place. Alright, so gases can also needs to move through here, and they will then also store some water and some starch. Then we have the endodermis, so that's a layer there just above that vascular bundle, and the endodermis in this case stores starch, and it can also be known as a starch sheath. And then the last thing we found in the cortex is the parenchyma tissue, and this tissue, as you can see from the image, they have this dark band that surrounds them, and that is basically just their cell walls that are extra thickened. They're thickened all the way through evenly, and that provides some support and protection for my stem. Right, so Yolandi will carry on with the vascular tissue.
Okay, so if we look at our center, this is now where all our Easter eggs are located, and they represent our vascular bundles. So by the time that our water or our nutrients, it's now been deposited into the xylem and the phloem, this is the highway through which these nutrients and water will move. Okay, so we're going to start off by looking at the vascular bundle and what it consists of. So we've got xylem on the inside, and I always make the association X marks the spot. So that's in the center you will find your xylem, and that was true for the root. And in this case, we find that xylem is the closest to that center. So this darkened layer of cells at the bottom will be your xylem. Your phloem will be on the outside. Okay, now you will notice that there's this dotted cap on top of it, and that represents your sclerenchyma tissue. Now their function is firstly to protect the vascular bundle and also to strengthen and support the stem. So quite often we see that people go out to trees on the street, and they actually remove the outer layer of bark, and if they take off too much of the bark, they reach that layer of vascular tissue that's surrounding the outer part of the tree. So if you literally remove a whole ring of the outer tissue of a tree, you are cutting off the water and nutrient supply to a tree. So you're actually killing the plant by making that layer surrounding it. So you have to remember that the vascular bundle is like the plant's vascular tissue. So that's their form of capillaries, arteries, and veins. Now, if you remove that, there's no way for the plant to actually sustain itself. So it's super important that they are protected. And as we've seen with that outer cortex area, with the cambium inside it, with the sclerenchyma cap, it all plays a role in ensuring that if, for instance, humans or animals start gnawing at the tree, that there is some form of cell protection before we get to the delicate tissues that are your xylem and your phloem. Right, so let's dig to the next part, which is the cambium. So that's a strengthening tissue, one of the strengthening tissues that we find in trees, and we find that in between your xylem and your phloem. But it also plays a role in secondary thickening. Now, I'm sure that all of you have seen a tree that's been cut through the middle, and they've even told you can determine the age of a tree by looking at how many rings are present within the tree. And now that is your secondary thickening that has taken place over the years, and the tree actually grows sideways, so it pushes outwards as it gets older and older and older. And we can determine how good a year was in terms of rainfall and how well nourished the tree was within a particular season by looking how thick those secondary thickening bands are. So a tree can actually provide us with a lot of information as to what the climate was like within its lifetime when we look at that secondary thickening. Right, so there's our sclerenchyma cap. We can see the phloem, we see the xylem, which is on the inside there, and then the cambium separates the xylem and the phloem from each other. Right, so let's take a closer look at that. Here we have our sclerenchyma cap at the top, so nice specialized cells, very little intercellular space, densely packed, no intercellular spaces in actual fact. We can see where the phloem is located here, that layer of cambium, and the xylem just beneath it. Now your teacher might give you a picture like this, or if it's a practical situation, or if they want to extend you a bit, they can give you a micrograph of the plant tissue. Now if we just compare what we see in the diagram to the real life situation on the right hand side, we start off with the sclerenchyma, which is at the top. Now see if you can identify where the phloem is in this diagram. Okay, there we go, second layer, cambium layer quickly. There we go, thin layer in between your xylem and your phloem. All right, fantastic. So that could be something that you might need to be able to identify. Right, so then we get to the center component, which is considered the pith. And here we've got loads of parenchyma tissue, so loads of storage cells that will obviously contain our starch. Okay, so it plays a role in storage and transports of substances. So if we have, for instance, sucrose, which needs to be transported to the cells, or starch, or whatever the case might be, glucose, then they can move across into this central area, which is the pith, where the parenchyma storage cells are located. Right, in between our vascular bundles, we've got medullary rays, and that's just pretty much extending from the pith outwards through those vascular bundles. Okay.
All right, so just quickly looking at a practical investigation that your teacher can also sort of use to extend your knowledge. So in this investigation, we can see or investigate whether water is actually transported through the xylem. So here we have a plant, and they should have also then just indicated that this plant is placed inside of water, and this water contains like a dye. Let's say in this case it has a red dye in there. Okay, and this water and the dye will now need to be absorbed by the roots and then transported through the stem all the way so that it eventually reaches the leaf. So then if we take a cutting from one of these leaves, a cutting through a root and a cutting through the stem and then through the root, this is what we'll basically see as a type of a diagram. So this is the stem and this is the root, and then the areas that contains this dye will then stain the xylem red. So in this stem area, the xylem being in the middle, it will then stain the xylem, which transports water. It will stain them to a red, to the same color as the red dye. And the same as in the root. So as Yolandi taught you, the X marks the spot, so that center area would then be stained red as it transports the water along with the dye. So this is an actual image of celery, which is a stem, and there you can also see these red little spots there, and that would then be part of the vascular bundle being the xylem, if we could zoom in on that and actually determine whether it's the xylem or the phloem. But that's quite a nice image to see these, a part of the vascular bundles inside of this celery stem. And then this is just a cutting from the celery, and then in some areas you can see there's this red dye on the inside, and there is another tissue, not certain if that's the phloem or maybe the sclerenchyma, but that was not stained. So this inner area was stained, being the xylem, and this then proves that water is actually transported through the xylem tissue. So quite a nice exercise for you to do is to maybe just put a piece of celery in a beaker of water and add some food coloring to that.
I've actually had a very interesting experiment that I did with my own students once. You take a rose and then the stem, you cut it into 4 sections. You actually take a scalpel and you cut through it in half and then in half again. And then you've got these 4 points sticking out. And if you put each point into a different color food coloring, it draws up all that coloring into the petals. And eventually you've got this rainbow rose that is formed with different colors, and it's the most beautiful thing. So definitely it's something interesting that you can go and make at home and then go and show off to your friends, like, look at this special rose that I, got. But in actual fact, you dyed the rose. So yeah, something to try at home.
Okay. Yeah, so quite an interesting thing of really visually seeing this process in action. All right, so digging back into the work, next up we're going to look at transpiration, and this is going to be the next sort of hot topic for this day. So just in terms of what is transpiration, there we have this magnification glass, not that it's possible to see these little stomatas with a magnifying glass, but transpiration is when water, that is indicated by these little squiggles, water that is lost through the stomata, and it is water in the form of water vapor, not water as in water droplets. And transpiration, it needs to happen, but the way in which transpiration, if it happens at a faster rate or a slower rate, that can be influenced by many things, which Yolandi will explain to us in a little bit. So this is a leaf. Hopefully you have learned about this in this term. So there at the top we have epidermal tissue, right? These brick-shaped cells, and we also have a cuticle there at the top, and then at the bottom we also have an epidermal layer, right? And then here you can sort of see this is sort of now where the water and the food needs to be transported, and here at the bottom there are these stomatas through which water needs to leave. So this leaf is adapted in many ways for transpiration. So it's adapted in a way for transpiration to happen at a sort of a moderate pace, so that it doesn't lose too much water and eventually just desiccates. So the way in which it is adapted is, firstly, there's quite a nice thick cuticle over the epidermal tissue. It's actually part of the epidermis, and this reduces the amount of water that is lost. Right, then on the lower side there's actually more stomatas on the lower side of a leaf compared to at the top, and this is quite a very smart adaptation because the lower side of the leaf isn't exposed to so much sunlight, so it's actually mostly in the shade, and being in the shade it won't lose as much water through transpiration. Just like you sweating more on a hot day in the sun versus in the shade, you'll sweat less in the shade. This plant will transpirate now less, having the stomatas on the lower side being in the shade. Then the next adaptation is actually the size of the leaf. Not sure if you've actually realized this, but all of these succulent plants, these plants that live in very, very dry conditions, their leaves aren't very large, right? And the size of the leaf sort of will also influence the amount of water that can be lost. So if the leaf is larger, then that means it should have more stomatas compared to a smaller leaf. So larger leaves will therefore lose a little bit more water compared to having a smaller leaf. So these are 3 ways, we'll look at 3 more, in which the leaf can be adapted for transpiration. So the next 3, there can be some hairs on leaves. If you've sort of felt some leaves and they are quite smooth, the hair is actually quite a nice adaptation, as it traps some of the water vapor, and that leads to basically less transpiration. So the way in which that is done is it decreases the diffusion gradient. So it makes it possible for less water to actually get lost. Another thing is there can be thorns. And the thorns actually take the space of where stomata could have been. So that if there's now fewer stomatas and more thorns, it means that there's less transpiration. And then the last thing is that some plants, especially succulents or cacti, their stomata is sunken. And I'll show you a picture in a moment of what this looks like in a microscope. But the sunken stomata actually it traps the water. So it sort of keeps the water inside so that less water is lost through transpiration. So this is a micrograph showing you a sunken stomata, and that little thing there, oh, my mouse is that little there we will have 2 guard cells forming a stomata. So you can sort of see there's this little trap that is formed so that less water can actually escape. And here also, there at the top, you'll see 2 guard cells so we call this entire structure there a sunken stomata, where the stomata is actually supposed to be sort of in line with the epidermis. There's not supposed to be this hole and the stomata forming there at sort of inside of the leaf. So those are a few ways in which the leaf can be adapted for transpiration, and then different plants, depending on their environment, they will then either lose more water through transpiration or lose less water. So different plants are adapted differently for their situations, and these are just sort of 5 quick ways of how plants can be adapted for their way of losing water.
Okay, so we're going to now look at the factors that influence transpiration. So these are external factors, which means environmental items that can result in a plant losing water more due to the process of transpiration. So if you think about it, what could potentially be, one of the items that will pop up is probably temperature. That's the first thing that comes to mind. So on a very hot day, we normally find that the plant will transpire more than on a cool day. And I'm sure that you have seen this yourselves. If you go into the garden and it's been a particularly hot summer's day, then your plants just look like, you've reached the last lesson of the day, and that plant also looks, a bit verlep, if you want to put it that way. So temperature plays a very important role in how much water the plant is actually taking up all the way to its leaves. And if it's lost too much water to the environment due to transpiration, then your cell is no longer turgid, the cells become flaccid, and then soon after, the whole plant will start to droop. So then it's an indication that maybe you should go and give it some water and try and save it. Okay, so that's our first item that we have, is temperature. Then we're going to look at the next one, which is light intensity. So obviously, if it's now overcast or whether it's a nice sunny day, that will influence the rate of transpiration. Humidity, if you're living in Durban, you will know all about humidity. It's when it's like really warm, you're sweaty, but that water's just not evaporating off of your skin because your environment has a lot of water particles in it already. So that's also going to affect your plant. And the last one we're going to look at is how wind influences the rate of transpiration. So I always see these trees being transported on the back of bakkies, and it's just leaves going at the back. And unfortunately, what happens to a tree is it increases the rate at which transpiration occurs. So now they're transporting it to its new location, and if they don't water it when it reaches its new location, that plant's actually going to die because it's now absorbed all of the water that's in the soil container that it was being transported in. And yeah, it's probably used up all of it depending on how far it's been traveling. Okay, so under normal conditions, the plant obviously has its water that is surrounding its roots, and the plant is capable of regulating how much water it's losing to a certain extent. But unfortunately, these factors, if they're all at play, will just speed up everything significantly. Right, so let's move into this section of the work quickly, and the first item that we will be investigating is the effect of temperature. Now, temperature has two roles at play here. One is that an increase in temperature means increase in kinetic energy. So the water particles are moving a bit more than what it normally would have. But we also find that water potential gradients are affected. So within the plant, you will have a lot of water particles. And if it's a particularly hot day outside, there will be fewer water particles. So already there's a driving force for the water to move from an area of high water potential to an area where there's a low water potential on a hot day. Right, but then knowing temperature, increase in temperature, we also find that the kinetic energy of these particles increase. So they start bumping against each other and they move more freely. So now, on top of the water potential gradient that exists, these particles are moving more freely out of the stomatal pore that we see here. Right, so if we just quickly do a run through it, an increase in temperature means an increase in kinetic energy. Here we can see that kinetic energy with the blue dots bumping against each other, that's your water particles. Okay, they move faster. This results in them bumping into each other more frequently. And if they bump each other just past that one cell, they're out of the leaf and gone. Right, so that will end up in them escaping from our leaf, and that's it for the water particle. Right, but what happens on a day when it's cooler? So tend to have less evaporation happening, you have more water particles in the air, but also the kinetic energy in low temperature conditions will mean that they are more sluggish. They will less frequently bump into each other, and less water particles will move out from the inside of the leaf. Right, so then as a result of it, we find that our plant is not going to lose as much water on a cool day. Okay, so then the question is, how does light intensity affect our plant? So on a sunny day, we've got an increase in light intensity. The light's really bright. We also find that there's an increase in temperature. And together, both of these items means that we're going to have an increase in photosynthesis taking place. Due to this, our stomata will open. But the question is, why are these stomata opening? How is it possible that photosynthesis directly links to stomata opening? And you might have been taught in your classroom that those guard cells that look like two kidney beans, they actually have chloroplasts inside of them as well. So when they photosynthesize, they actually start to swell up to a certain extent, and they open up the pore when the light is present. So now, because that opening is nice and wide, we find that there's an entryway for the water to move out, and we now have an increase in the rate of evaporation, loss of water, which we then call transpiration. Okay, and that's obviously what we see within that last step. We have an increase in transpiration. So the question, what happens on a cool day when it's overcast perhaps? We're going to have the exact opposite. Light intensity will be low, temperature will be low, due to the lack of sun, because it's really overcast, will have less photosynthesis. Stomata would not be as open as it used to be, and that rate of transpiration will also decrease. Okay, so now the next one that we have is wind. How's wind going to influence the sky? And yet again, water molecules, they move from an area of a high water potential to an area of a low water potential. Now, if the wind is constantly removing a layer of water on the outside, naturally the water molecules will move from that high area to an area where there are fewer particles. They are now also bumping into each other, and we find that they're going to move out through the stomata. So here we show that there's an increase in water vapor concentration on the inside, meaning a high water potential area, to an area where there's less water vapor, low concentration, because the wind, as those water molecules are coming out, the wind's taking it away immediately. And that is an increase or a massive drawing force for water to constantly be removed from that leaf. And as we've already seen, in the case of where the wind's been blowing for several days, maybe by the third day your plant's no longer, they're no longer looking too healthy. Right, so just to run through it, an increase in wind means that our water vapor is being blown away from the leaf. This results in a higher concentration gradient of water vapor particles, and water escapes through the stomata. Now, when the wind is not blowing as fast, it's the exact opposite. So now the water concentration's not going to be, gradient's not going to be as high. So less water is going to be taken away. This lowers that concentration gradient, and less water is going to escape from our stomata. Right, the last one that we have is humidity, the last one. And the example we're going to start off is in a case where there's low humidity outside. Okay, so this is now where there's a lot of water particles on the inside and very few water particles on the outside. That's not typical summer's day in Cape Town. And we find that the water molecules will move from an area where there's a high concentration of water vapor molecules to an area where there's a low concentration. Okay, so that would mean that outside of the leaf, we're going to find that the water is going to move out due to that concentration gradient that has been established, and they escape through the stomata. In terms of a high humidity, this is now a typical Durban situation where we've got a lot of water particles in the air, and compared to on the inside, that gradient's not going to be as steep. So going through it, high humidity on the outside means a high concentration of water vapor on the outside of the leaf. So these water molecules are already within the air. This lowers that concentration gradient, and less water is likely to escape from the stomata.
Okay, so next up we'll be looking at how the water is now actually transported through the plant. So as we can see, water lands here in the root, in the soil, and then the water needs to basically be absorbed from the roots, and then from the roots it needs to travel to the stem, and then eventually to the leaf, and then from the leaf the water vapor will be lost through transpiration. So looking at this in a better image, we have over here we have the root. So this section over here illustrates the root. Over here we have the xylem of the stem. So xylem because we're looking at the transport of water, and then eventually it reaches the leaf. So there's actually 4 pathways that the leaf has, that the water needs to travel through. So it'll firstly it needs to be absorbed, right? So it needs to be taken up from the root, and there's a whole process involved there. Then it actually, as it enters the root hair, it then needs to travel through these parenchyma cells until it actually reaches the xylem. And for that, there's also a process that takes place. And then when it reaches the xylem, it needs to sort of, as you would suck through a straw, the water needs to be sucked up against gravity, because the leaves are obviously on the top of the plant. So then it needs to move up through the leaf, through the stem, until it eventually reaches the leaf. And then the fourth way is the water now needs to leave the leaf through transpiration. Okay, so we'll be looking at these 4 pathways. So the first one would then be the absorption of water, and this process is actually only due to osmosis. So in terms of how this works, there's actually a higher water potential on the outside of the root, and there's a lower water potential on the inside of the root. So this root actually also has a vacuole. Not sure if it's indicated in this image. Let me just take that away. But there's a vacuole inside of here. The vacuole is filled with cell sap, and the water potential inside of that cell sap is now is lower compared to the outside. So due to osmosis, the water will basically move into the root from the soil, and then it basically moves into the vacuole. The vacuole can swell up, and that then leads to turgor pressure. Alright, so the mechanism through which water moves into the root is then osmosis. Okay, and here you will find just a short definition of what osmosis is. It is the movement of water from an area of high concentration, so in this case it was outside of the root, to an area of lower concentration, a lower water potential, through the semi-permeable membrane. So now from there it needs to move through the root until it reaches the xylem. Right, so now we've covered this first part, moving over to the second part. So here it has already now been absorbed through the root hair, and now there's actually a few ways in which the water can now travel. The one way is a bit of a more difficult way, as it's the main process, but it takes a little bit more time, since the water needs to pass through the cell wall, through the cell membrane, into the cytoplasm, then out of the cell membrane, out of the cell wall, and then into another cell. So there are many membranes that it basically needs to pass, and that is one way in which the water basically it squishes through into the cells and then into another cell. The quicker way happens in this black pathway over there, where it sort of just moves along the cell walls. So it doesn't pass to the inside of the cell, it just moves here through the fusion, along the cell walls into an intercellular airspace, and those are basically the 2 ways in which water can move until it reaches the endodermis. Then we have a different story. So the reason why I mentioned that is because this endodermis has the Casparian strips that we mentioned right at the start, and that basically forces the water to enter the cell wall, and that basically acts as a type of a bodyguard that only allows the water to pass through and not any toxic substances. So then from the endodermis it will move through the pericycle, and then voila, it finally reaches the xylem. Right, and that is then the happy day, because now the water can finally be transported from the root to the stem. Alright, so those are then pathway 3 and 4, which we will just quickly run through, which is basically these 3 processes. root pressure, transpiration, pull, and capillarity.
So the three that we have on the board at the moment, the very first and most important drawing force that we have is transpiration pull. So if we go to the next slide quickly, just to explain what it entails, is you have transpiration, which is the movement of water molecules from the inside of the leaf to the outside environment. So that's called transpiration. It is due to the fact that you have evaporation of water particles. So the question is, what's the difference between transpiration and transpiration pull? And the answer comes in that due to the fact that we are losing the water to the environment, water needs to replace that area. So water is continuously moving from an area of a high water potential, which is inside of your xylem, to an area of a low water potential, which is here inside the leaf. So the question is, what created that low water potential? It's because transpiration has occurred. Now that on its own means that you always have this drawing force, upwards drawing force against gravity of water particles. So the question was, how do we have that suction that occurs? How does water move against gravity? Transpiration pull is one of the answers. So there we see water being lost to environment, diffusion, evaporation is occurring. We call that transpiration. Okay, so here we've got just explaining, going through the mesophyll layers, eventually moves out of the leaf, and then it's gone to the environment. The whole process of drawing the water out, we then refer to as transpiration pull. So here in the image, it shows it nicely. There's this continuous movement of water particles upwards because it's replacing the water molecules that has been lost to the environment due to transpiration. So that's the main suction force that we were referring to. It's always in an upwards motion because where do we lose water from? We lose it from the leaves. Okay. Then the next ones that we have root pressure. And water particles are continuously moving into our cells, and then also at the top we are continuously losing water to the outside environment. But it's almost as if the water molecules are pushing up against each other. So that pressure is coming from that continuous flow of water into the root from the soil. So obviously, you know, the water needs to go somewhere. The only place that it can go is upwards against gravity. Okay, in this picture we can also see the water molecules, it's kind of clinging to the sides of the stem. That's another force that's at play here, and we refer to that as being capillarity. Capillarity is divided into two different forces, which is adhesion and cohesion. So adhesion, that's the attraction of water particles to the walls of the xylem. So here we can see it's attracted to the walls. Cohesion refers to attraction force between water molecules itself. Now we normally see that on a car. If you're sitting outside and it's raining and you just parked in a parking lot, you see two water molecules coming close to each other as it runs down the window screen, and the next moment they're so close to each other it forms a big water droplet. That is referred to as cohesion. So together, those two forces allow for the movement of water throughout the stem as well. So that brings us to the end of the forces at play and our chapter for today. So it's a mouthful. I know it's a lot of work. We wish you the very best for your upcoming tests and practicals, and hope that this has helped you understand the concepts a little bit better. So all of the best.